Darwin Day. I'm Eric Selger, President of the Red Bank Humanists, and thank you all for coming today. So, I've gotten a request from our speaker, Ron Flannery, to be brief, so I won't do a long introduction. Many of you know him. He's one of our Board of Directors members, and okay. So he's going to come and talk. He's going to step up and talk to us about bioethics and evolution in the 21st century. So everyone, a warm welcome for our own Ron Flannery.
Happy Valentine's Day. Thank you. Okay, bioethics and evolution in the 21st century. A couple of speakers' notes before we get into this. Please save your questions for the end. I've got a lot to say, and I'm only going to say it once. But I do have a lot to say, and not a heck of a lot of time to say it, so I hope to get it all out there. My slides and my notes are available. My email address is webmaster@redbankhumanist.org, because I am the webmaster at redbankhumanist.org. And if you send me an email, I will send you a link to download all this stuff. But you got to promise me if you download it, you have to read it. And there's a lot of it. So when I put this presentation together, what I wanted to do was organize my thoughts, and I did that by coming up with a list of questions. These are my questions. And we're going to revisit all these questions again at the end. But this is basically an outline of my talk. First of all, is evolution still relevant? Because clearly we're still evolving in that mutations constantly occur, but does natural selection even play a role in that anymore? And if we have the technology to start to control our own evolution and the evolution of the biosphere that we live in, should we do that? Because there are many, many implications around that. And then if we do decide it's okay to do those things, where do we draw the line? Is it okay to do it with plants, with animals, with humans? Is it okay to change just a person but not their offspring, and so on? So that's the foundation that we're going to lay, and then we'll have a discussion at the end, and then we'll decide for what that's worth. So what is bioethics? Well, scientists are constantly discovering things. Our knowledge of biology and science in general is growing at about 40% per year, which means in less than a year, or less than 2 years, about a year and 9 months, I did the math the other day, our knowledge of science doubles. And at the rate we're learning things, it's pretty clear that we have to start thinking very quickly about what are the implications of the things that we're learning how to do before we start doing them. And it's not just doing them out in the real world. What's it okay to do in a laboratory, for that matter? So now I'm going to teach you how evolution works. And if you've heard all of this before, you can take a nap and I'll wake you up when it's over. But basically, a cell is a factory. And that factory is used to modify a living thing and control that living thing. And that living thing is the cell itself. So it's a factory that runs itself, and it's got little machinery inside it. And it also communicates with the factories that are around it. The factory, when it's generated, comes with a set of how-to books, because the factory has to know how to operate itself. So a human factory, a human genome, consists of 24 pairs of how-to books. And those are chromosomes. And the chromosomes are written in a code that's made up of 4 chemicals. If you were to stretch this code out, what you would see is that you've got lots and lots, oh, I can't stand there. You've got lots and lots of information in that genome. There are 21,000 chapters in these how-to books. And each chapter is how to do something. Now, the simplified version is, you've got a gene, that's a chapter, and it tells you that this is how you make your eyes blue. but it's not really that simple because one gene always, or almost always, affects multiple other genes. So they're all tuning each other. So it's really a lot more complicated than that. But just to keep it simple, you've got about 3 billion letters of information in your genome, and that genome contains all these instructions. So when the factory wants to do something, when it's time to do something, like you've got a stem cell, and that stem cell has to become a nerve cell or a liver cell, how do we know how to do that? Well, what happens is that instruction has to be read by a piece of RNA. So there's a machine made out of RNA that comes along, reads the genome, and broadcasts the instructions through the cell in the form of proteins. So what we've got then is genes directing the production of proteins. The proteins do most of the work. They tell the cell what to do and how to do it and how much to do it. So proteins don't just appear, they appear in quantities. So more of a protein is going to have a bigger effect than less of a protein would. So if you change or mutate a gene, here's the test question, right? If you change or mutate a gene, that's going to cause the expression or the repression of one or more proteins. They might be new proteins, they might be proteins that we've had for 1 million years, but changing the gene changes that expression, and that change is going to impact the structure, the function, or the regulation of a tissue or an organ. Okay, so what does that all mean? Well, you've got some bugs living on the bark of a tree, and they're green. And 2 of these green bugs mate, and they produce offspring, and one of them has a mutation in the egg that gives it a brown carapace. And because it has a brown carapace, it's harder for the predators that feed on those bugs to see, and that means it has an advantage over its brothers and sisters in that it's harder to see, and it's likely to survive better than they are likely to survive. So that mutation is going to be selected for, not in any intelligent, thoughtful manner. It's just a matter of luck, right? That beetle was lucky to be born brown, so the birds are eating all its brothers and sisters. That beetle gets to have children. 50% of them are going to be brown, or at least 25% of them are going to be brown, depending. And that means over time, there are going to be more and more brown ones and fewer green ones, and eventually that species is going to be brown instead of the green it used to be. Okay? Pretty simple, really. But natural selection in total gets a little complicated. I'm going to keep it as simple as I can for you. I'm going to give you 4 rules. Natural selection requires a variety from which to select. So where does that variety come from? Well, there are abrupt changes that are caused by mutations, and there are slow changes that are caused just by variation, and I'll talk a little more about this later. If there are no new features introduced, though, if there is no variation, if there is no mutation, then there is no evolution. Natural selection also requires the ability to produce viable offspring. So if there is a mutation that makes an insect fly faster than all its brethren, but it can't lay eggs anymore, then that's a pretty useless mutation. It's going to end when that germ dies out because it doesn't reproduce. So not only must you have offspring, but they have to survive long enough so they can reproduce. So if you get a mutation that produces too few offspring, or they are easy prey, or they're subject to diseases that prevent reproduction, the germline dies out. Again, no evolution is happening there, at least not a positive evolution. And then the offspring have to pass on their higher survivability traits. And the more offspring that pass on the high survivability traits, the more likely the survivability of the germline. And that passing on of these traits through the population has a name, and it's called population sweep, I think. Okay, so now we get to the point of how is evolution working for us? Well, about 6 million years ago, hominids and chimpanzees diverged from the primate line. So anybody that tells you that we evolved from chimpanzees is not telling you the truth. We both evolved from a common ancestor who is now extinct. About 3.5 million years ago, a gene called SRGAP2 got duplicated. And at that time, the size of the hominid brain went from about 350 cc's on average, or cubic centimeters, to somewhere between 400 and 500 cubic centimeters. So this was a pretty big leap. That was a leap of, around 25% in the size of the brain of the hominid. Still pretty small, but an amazing increase. Then a million years after that, the same duplication occurs again. We get Homo habilis with a brain of 600 cubic centimeters. A slightly smaller increase, but still in the 20 percentiles. And that increase in intelligence gave us the use of stone tools and a meat diet. So when you read some papers from anthropologists, they'll tell you that changes in behaviors cause the brain to increase in size, but in reality, I think it was exactly the other way. About 2 million years ago, another gene mutated, this ARH-GAP11B, had two effects. Again, it grew the size of the brain to 850 cubic centimeters, and look what's happened over about 1.5 million years. You see the brain has just about doubled in size in the hominid line. And also at this point in time, this convolutions in the surface of the brain appeared. And it's those convolutions that give us a large surface area to our brain, which is unique to humans, and that gave us Homo ergaster. Quickly followed by Homo erectus, the crafting of stone tools, the use of fire. And you'll notice at 2.5 million years ago, there was the use of stone tools, but those are naturally found objects that were used just because they happened to be the right shape and size. But with Homo erectus, the stone tools were actually starting to be formed with two sides and two edges, clearly showing the sign of intelligence. Now, about 1.2 million years ago, it's estimated that the total population of hominids was only about 55,000 individuals. So imagine what happens when there's a mutation in a population that small. It's going to sweep through the population pretty rapidly. It's not going to take a lot of generations. So these changes that we see in the evolution of hominids, because the population was as small as it was, are pretty rapidly spread. About 150,000 years ago, another major change occurred in the now human brain, and that was the enlargement of a segment of the brain up here called the precuneus, and this is where visual integration takes place, and it's considered the seat of all self-regulated activities in the brain. So when you do an Einsteinian kind of thought experiment, that's the part of the brain you're using. You're visualizing things and imagining how they would work out. And you're also imagining how you fit into society in that part of the brain. And again, this is something that's peculiar to humans. Then about 70,000 years ago, there was a big extinction event. A major volcano exploded. And we went into an ice age for 1,000 years. It's estimated at that time, I've read several articles that have the number even lower than this. They have it around 2,000 individuals. But there's somewhere south of 30,000 individuals alive in the world. Another evolutionary step, a mutation again, occurs 50,000 years ago. The FOXP2 gene gave us, and this is an educated guess, that's where we got speech from, the evolution of this particular gene. And also what was happening then, this is after the Ice Age, humans, Homo sapiens, started to meet Neanderthals in Europe, and there was some interbreeding that took place. And that gave us an advantage in that we picked up a lot of evolutionary advances that the Neanderthals experienced and gained over their thousands of years by interbreeding with them. It's estimated today that everybody in this room has from 1 to 4% Neanderthal DNA. Anyway, this all resulted in something called the Great Leap Forward, where human culture suddenly starts to accelerate very fast. People began doing things that show that they are starting to communicate and cooperate, because they're starting to do rituals, and they're burying their dead and wrapping them in skins and burying them with things that they owned, artifacts. They started to create clothes from animal hides. They start making weapons out of bone, so they're much more dangerous than they ever were before. They invent the flute at this point in our history. And they develop complex hunting techniques that, again, require participation by large numbers and cooperation and communication. Then something really amazing happens 20,000 years ago. And to me, this is a turning point in our story, so try to understand this one. The human brain begins to shrink. And when you do research on this particular topic, why did the human brain begin to shrink? You read a lot of very nonsensical theories. The one I like, which I do not think is nonsensical, is that at this point in our history, we started to do directional selection. Instead of natural selection telling us what we were going to become, we started selecting each other. So we might see someone across the room and think, wow, that girl's got talent, you know, and I'd really like to get to know her. And this was something that was kind of new because talent was a new thing. So people are starting to select themselves for social characteristics. Happy Valentine's Day. And this is called self-domestication, because that's what's going on. We're starting to look at people and saying, well, you know, I like them young. Here's somebody who looks young, right? And that's something that goes on today. People find younger looking people attractive. Women find tall men attractive. Men find women with large variation in their dimensions more attractive than women that are straight up and down. And that's all a result of this kind of thinking. So the human brain begins to shrink, and self-domestication, or domestication in general, has a couple of interesting side effects. One is a smaller brain. If you look at dogs compared to wolves, a domesticated animal, they have smaller brains. They have smaller bodies than wolves. And that's called neoteny, okay, because you tend to stay more childlike when you've been domesticated. And it's seen in the skeletons of humans through that period of history. Another big thing happens. Another mutation occurs. The ASPM gene, which impacts neural development, mutates, starts producing some new amino acids that it never produced before, and we get another big leap forward. We get agriculture. We get invention of things like irrigation and deforestation to promote the growth of plants that we prefer over plants we don't want. We get animal husbandry, food preservation, and written language. So we've had written language going back to this particular mutation. At that time, the population now of humans is estimated to be about 4 million people. Still not a heck of a lot. So a sweep of a modification to how you look, how you behave, how you think, is going to move through the population pretty quickly. And also the way we lived changed drastically at that time, because when you go from a hunter-gatherer society to an agrarian society, a lot of things change, right? You're not moving around anymore. You suddenly have to deal with things that you never even thought were problems, like sanitation, right? Where do I get clean water to drink if I don't move around anymore? And where do I bury people? And how many people can I bury? And these are things that require some thought. And these are all new, brand new thoughts. So we're going to have to develop some kind of civilization in order to deal with all of these issues. And that's what starts to happen then. You get the invention of laws and the rule of law. And that changes the whole concept of natural selection, because now it's people who are saying, if you behave in this way, we're going to remove you from our society. But if you behave in this way, we're going to promote you in our society. That's not natural selection. We invent things that never existed, like monogamy and arranged marriages. And we're really into selective breeding at this point. The rich are marrying the rich. The powerful are marrying the powerful. alliances are being formed, right? Forget all about natural selection at this point. We're inventing armies and war and things like genocide and actually carrying them out. So natural selection, as a working machine, is starting to get interfered with in a major way. We moved from a majority rural society 100 years ago to a majority urban society that we live in today. We get crowding that affects sexual orientation and libido. We invent something like the pill, which gets in the way of how birth rates work in a natural environment. And we start to get exposed to chemicals whose exposure we don't even understand a lot of times until after we've seen the effects accumulate. And now we know that through a mechanism called epigenetics, some of these effects don't just affect us, they affect our children and our grandchildren for generations. So sometimes, we'll do something to ourselves, we don't know we did it, and then it impacts future generations. Epigenetics, by the way, is a tweaking of how the genes operate and how much protein they express based on external factors. There's a whole discussion we could have about that topic too. So let's look at natural selection over time. We start out hunter-gatherer society. Now one of the things people talk about when they talk about natural selection is lifespan. So lifespans we know for sure have been consistent over the past couple thousand years, and probably going back 10,000 years or more. People have been able to live over that entire period of time for about 80 years. What varied, though, in a big way, was average lifespan, because average lifespan is affected by whether you even survive birth, right? If you die at birth, if half your population dies at birth and the other half lives to be 70 years old, your average lifespan is 35 years. Also, you can be killed by animals, you can be killed by accidents. We don't have real doctors in a society like this. We've got witch doctors who, you break your leg and they do a dance and a song, and hopefully your leg will heal straight and you'll be able to walk. Well, good luck with that. And if you get an infection, they don't have antibiotics, you're probably going to die from that infection. There's tribal warfare. And it's kind of small stuff, but you can still get killed. The good news is, you might be able to survive childbirth, and if you do, then whatever enabled you to survive that childbirth, maybe your head was just a little bit smaller than the kid next door that died, or maybe you had a little bit better immunity built up by your mother while you were gestating. So that's going to be selected for because you survived. And if your mother survives childbirth, that's going to be selected for whatever enabled her. Maybe she had wider hips, and your big head managed to get through after all. So those kinds of things are going to be selected for. And if the mother survives, she'll get to have more kids, and whatever enabled her to survive will get passed on. And if you survive, you'll be able to have kids and pass that on as well. Also being selected for are the intelligence and social skills that allow you to avoid being killed by an animal or dying in an accident or getting killed in tribal warfare. So the ability to avoid predators, use shelter, use fire, make weapons, hunt in groups, cooperate with your fellow men, these are things that are going to be selected for. Then we move into an agrarian society. And remember, now we have to worry about basic sanitation. So what are people dying of now? Well, they're still dying in childbirth, not as much maybe, but now we're also getting hit with things like cholera and smallpox and rheumatic fever. Bear in mind, in Europe, everybody in Europe goes on vacation in August. And they do this for historical reasons. Because if you hung out in London in August, you were going to get the plague. Because it got hot in August. And everybody was throwing their sewage into the river. So it was a good idea to leave town, head for the country, and wait till things cooled down, then come back to the city. Other things that were happening, people are dying of infections in childhood and after accidents. People are dying of parasites that they never even experienced before. It used to be thought that people in the South were lazy. Turned out what was really happening is people in the South were working in the fields And they would, when they had to go to the bathroom, they'd go off to the side and they'd do their business and they'd come back. And they weren't burying the stuff, it was just there. Well, there's a parasite called pinworm that can crawl about 6 feet. So you wouldn't go right where your buddy went, but you'd go near where your buddy went, you'd squat down and you'd get his pinworm. And then you'd spread it to all of their friends. And pinworm would sap your strength and it would make you act as though you were lazy. Right? So this laziness was spreading throughout the South, and everybody in the South was considered lazy until somebody invented the outhouse, and they figured out if you dug a pit 6 feet deep, the pinworm would go away with a little help. Another thing that might be selected for, though, besides being able to not accept parasites, is maybe living symbiotically with them. So there are a lot of parasites that actually have beneficial effects to humans, because that's something that was selected for over time. And again, intelligence and social skills and cooperation are things that are going to be selected for. When we move into the industrial age, doctors actually started to appear on the scene who didn't kill you outright, but sometimes they could actually save your life. And the average lifespan went up to 45 years. And then we moved into the modern era. And if you look around now, what you'll see is people are dying of old age diseases, primarily. So coronary artery disease, stroke, cancer, diabetes, dementia, things that happen to you late in life. So here's the million-dollar question: what are we being selected for now? Being born with an IQ of 180 or being born with an IQ of 90 doesn't really make a difference on your ability to survive today. Being able to run faster doesn't make any difference, right? So what kind of mutation could you possibly have today that would be selected for in the light of what's happening in our society and in this culture that we built up? So next question on my list was, what are we going to do about it? Should we do anything about it? Can we do anything about it? So let me tell you about Craig Venter. You've probably heard of him. His name is pretty famous in this arena. In 2010, he did something really, really interesting and unique. He figured out what the genome was for a bacteria, right? So he took the genome of a bacteria out of the bacteria and mapped out what it was exactly. And then he took that information, put it into a gene sequencer, and generated an artificial copy of that genome letter for letter. And it was 580,000 letters long. And he injected it, using a very fine needle, into a cell. And that cell became a living, replicating bacteria. So he created the first artificial life form. Now, it was a copy of a real life form, but it was manufactured. And that had never been done before. So think of the implications of that. He manufactured a living organism. Then in 2014, Jeff Beck did the same thing, but he did it with a twist. What he did was he said, well, I'm going to take the genome of a yeast, and I'm going to figure out all the components of that genome, and then what I'm going to do is I'm going to throw away all the parts I don't think I need. So he went through the genome and he says, well, this looks like junk DNA, and he took that out of the mix. And, you know, this looks like something that might have been used once, but it's not being used anymore, so I'll take that out. And he cut the genome down to 274,000 letters, injected it into a yeast, and it lived, and it replicated, and it worked. So what did he learn? He learned, yeah, you can not only build an artificial thing, but you can, in essence, improve it. Now, he didn't engineer that thing, but what he did do was he said, these are valuable pieces, and these are pieces that don't look valuable to me, and he was right enough that the thing lived. So today there are laboratories that you can send a piece of material to and have them analyze that genome, that DNA, and you can send them a list of chemicals that you want mixed together in what order, and they can build one for you. And this is a very common thing. It's just 4 chemicals, and they have the machinery to do it, and it's not even all that expensive. Craig Venter has a new institute. It's called the Institute of Human Life, or Human Longevity, excuse me. And he will give you a complete and total breakdown of your genome for $250, and he can do about 20 of them a day. Okay, next major step in this process, though, is around 2010, Jennifer Doudna is a scientist who works with bacteria. She was looking at the impact of viruses on bacteria, and she discovered something interesting. When a virus invades a cell, it steals components from the cell to manufacture copies of itself. And a lot of times in doing that, it'll destroy the cell. So if you get invaded by a virus, what's happening is it's taking pieces of your cells to make copies of itself. And in the process, it might damage your cells and it might destroy your cells, and you get symptoms and you start sneezing and your body fights it and you get a fever and so on. But you survive. And eventually your body learns how to fight off the virus. But in a bacteria, it doesn't have the luxury of being able to do that stuff because it's a one-celled animal. It has to defeat that virus as quickly as possible and repair any damage that the virus made. So how does it do that? So Jennifer discovered that there's something called CRISPR-Cas9 that the bacteria invented, and it's a mechanism for repairing its own DNA. Pretty cool stuff. And she wrote a paper about it, and she said, gee, I wonder what the implications of this are. And that was read by Emmanuel Charpentier, who ran into her at a conference in 2011. And Emmanuel and Jennifer talked about it. They started to work together, and they produced a paper, a document that suggested here are ways you could use CRISPR-Cas9 to not only repair DNA, but maybe modify it in ways that seem desirable. Feng Zhang, who was an enterprising guy, read their paper, ran into his laboratory, and actually started doing what they had suggested and patented the process. And today we have CRISPR-Cas9 and variations over time. In 2016, they're going to advance that even further to the C2, C1, and C3 processes, but they're all basically the same in the mechanism. And I'll show you how that works. You remember this slide, right? Cell is a factory, instruction books, and you read the books and generate the proteins. Question is, how do you know where a gene starts and ends, right? That's a big part of the problem. If you're going to go in and modify a genome, you want to find the gene you want and only touch that gene, or maybe only half of that gene or whatever it is. So how do you find where the gene is? Turns out, in the genome, when a gene starts, there's a header there. And it's just a sequence of letters. So how do you even know it's a header? Well, you know it's a header because it repeats itself, and when it repeats itself, it does it in reverse. It's a palindrome. Isn't that amazing? And that clustered, regularly interspaced, short palindromic repeat is identifiable. And it's called CRISPR. That's where the letters CRISPR come from. So if you could take that repeating sequence and generate a guide RNA that lines up with it, then you could potentially find it, right? And then if you also take a genetic sequence and attach it to that guide RNA and wrap that around with another protein called CRISPR-associated protein 9, that's the Cas9 piece, now you've got a little machine, and what that machine does is it goes down the genome until it finds a match. And then the Cas9 piece says, okay, and I'm going to cut out what's there, and I'm going to replace it with this piece you handed me. This technology is very well tested. It's very inexpensive. I have read, although I haven't tried it out, that you can go online and buy everything you need to do this at home for $65. So here's the test question. CRISPR-Cas9, what is it? It says DNA has markers that say cut here, and we can make little machines that go in there and find those markers, and we can use the DNA repair mechanism that was invented by bacteria to edit DNA. We can add stuff, we can delete stuff, we can activate dead stuff, and we can tweak it. We can use the epigenetic mechanism to control how much stuff that gene does compared to how much it used to or how much we want it to. It's been used a lot in the laboratory on a lot of different kinds of animals. Like I said, very well tested. And it's even been used to cure some diseases that have been incurable forever. So it has big implications for humans. If you can cure muscular dystrophy in a mouse, you could probably do it in a human. If you can cure liver diseases in rats and pigs, you could probably do it in a human using the same technology. George Church is editing the genomes of stem cells before he turns them into nerve cells so he can try out different causes and cures for mental disorders. So tremendous potential here. Now we have to go back and talk about sex. But it's Valentine's Day, so not a big problem. Chromosomes come in pairs. Remember I said you have 24 pairs of chromosomes in your genomes. You really have 48 because you got a set from your father and you got a set from your mother. And I'm afraid to come over here. And your mother and father, they had 2 sets that came from your grandparents. So you have 4 grandparents, and you see you've got 4 distinct sets of chromosomes because of that. When a male produces a gamete, or a female produces a gamete, what happens is there's a random selection from in here of these 2 sets, and you get a mix. And this is how you get variation in a population, right? If you look at a family with 10 kids, you see 10 different people there. You don't see 10 copies of the same kid. They're all different. Some might be blonde, some might have red hair, some might have blue eyes, some might be taller, shorter, faster, slower. So back in the days when natural selection was actually going on, that variation gave you the ability to evolve slowly over time and select for things that were small changes. So mutations gave you rapid changes in people, where this kind of variation gives you slow mutations in people. Then when you combine the 2 gametes and you get your zygote, see, now you've got pieces of all your grandparents in here, right? And you're back to 2 sets. Now suppose, though, I'm one of these scientists that says, well, I want to produce a mutation here of my own making by editing this genome in this egg, and I want to pass that on to the offspring of this creature. Let's say this is an Anopheles mosquito that carries malaria. And we want to modify the egg of one of the mosquitoes, or a whole bunch of the mosquitoes, and give them a new property that says you can no longer carry the kind of malaria that impacts humans. And when you produce your offspring, that mutation that we just created is going to get passed on. The trouble is, when this offspring produces its own children, remember when it produces the sperm, it gets a mix of what was up here. So there's only a 50-50 chance that that modification you made is going to get passed on to that offspring. So half of the mosquitoes won't be able to carry malaria, but the other half probably will. Well, in 1957, biologists found an exception to this 50-50 rule. They found that there are genes that force themselves to be copied in with a much higher percentage of probability than 50-50. So instead of 50-50, it was more like 80-20. But what they suggested was, if you could take a mutation that was desirable and attach it to that gene that forced itself on, then you could force that mutation on as well. And that's called the gene drive. And the guy that figured this out suggested, his name was Austin Burt, he suggested that we use these selfish genes to do just that. And what he suggested was, what if you could take a malaria mosquito, you could attach that mutation to the gene drive, it would always get selected for, it would always get passed down, and when this guy goes up into that position, it'll be the same thing over and over again, right? So you can actually force this mutation to spread very rapidly through a population. And Bert suggested, just what I was talking about, that you use that gene drive to alter mosquitoes that spread malaria and stop the spread of human impacting malaria. Problem was, when he came up with the idea, we didn't know how to do this. We didn't have the tools to do this, but now we do. Now we have CRISPR-Cas9. So what happened was, in 2015, somebody followed up on Bert's work and actually did it. In the laboratory, we have constructed mosquitoes that cannot carry the kind of malaria that impacts humans, and we've attached it to the gene drive, and we could cure malaria in sub-Saharan Africa and save 1 million lives a year. We could do that. Recently, you've heard about this, the Zika virus. This Zika virus is carried by a mosquito, I forget its Latin name, it means African mosquito. But it carries other diseases too. Hydroencephalitis is a result of the Zika virus, but it also carries dengue fever and chikungunya. And this company, Oxitec, has modified, although I haven't found anything that tells how they did it, but I suspect they used CRISPR-Cas9 to do it. But they've modified that mosquito so it dies at the larval stage. So this is a pretty safe thing they feel to do, right? Because if you modify a mosquito so it doesn't carry malaria, there might be unintentional side effects that are going to get passed on and driven through the population of mosquitoes, and maybe they'll carry over to something else, and who knows, right? So it's something to fear. But if you generate a mosquito that's just not going to be able to reproduce itself, maybe that's a pretty safe thing to do and doesn't have too many unintended consequences or any at all. And if you test it in the Cayman Islands, in a place that's isolated, and it does go awry, well, you can go in with insecticides and kill off all the mosquitoes and save yourself from any unintended consequences. So that's what they did. And it turns out that they can reduce the mosquito population that carries the Zika virus by up to 96%. That's pretty powerful stuff. So they're thinking about doing it. They're going to potentially do this in Brazil because of the Olympics coming up there. They've got a lot of pressure to do it. And also in Florida because it turns out that dengue fever and chikungunya are prevalent in Florida, and they suspect, climate change may be a potential cause of that problem. So I think I've presented the case that evolution, natural selection may not be working for us anymore as much as we're doing it to ourselves at this point. And we've got the technology to really go in whole hog at this point as well. So we could really start changing ourselves and our world in some direction, for better or for worse. But then the question is, should we do it? And if we do it, where do we draw the line? Do we say, okay, we'll do it, but only with plants, or only with plants and animals, or only with animals that we don't use for food, or can we do it with humans? And if we do it with humans, do we do it so it only impacts a human and not their offspring, or it only impacts a human and 50% of their offspring, or do we attach it to the gene drive and drive it through the whole population? So let's look at some history. We started messing with plants 10,000 years ago, right? And the way we started doing it was we said, well, this is a good plant to eat, this is a good plant for medicine, this is a good plant to help things taste better. So these are plants we're going to cultivate, and because we're smart, we figured out how to cultivate them. And because we're smart, we figured out how to get rid of the plants that might interfere with that cultivation. And that led us to do selective breeding, right? We started selecting which plants we were going to breed. Pretty simple. But then in the 1800s, guys like Mendel came along and said, selective breeding is cool, but look at what you can do with crossbreeding. And remember, he did all those experiments with peas, and that's when people started doing crossbreeding and saying, okay, well, I like this plant, but this this variant of that plant is much more disease resistant. Let me see how many ways I can combine them and get the same kind of food I get, but with better disease resistance. And that kind of crossbreeding became de rigueur in farming. And that's when we started getting things like designer plants. So we've been monkeying with the genetics of plants for a couple hundred years now. Then in the 40s and 50s, something really bizarre started to happen. And I think it was a result of all those black and white science movies, science fiction movies I watched when I was a kid. They started subjecting the seeds of plants to radiation and chemicals, hard radiation and chemicals. And then they would plant them and see what happened. And most of them died. But once in a while, something new and different would come up, and people would look at it and say, pink grapefruit. Wow. And that's how we got pink grapefruit. And that's also how we got the semolina that's used in all the modern spaghettis that you buy. It was from radiated seeds. Then moving into the 80s and the 90s, people started thinking about actually editing the genetics of plants. And they didn't have great tools to do it. They would take a gene from a fish that they thought might benefit a corn plant, and they would attach it to a virus, and then they would infect the plant with the virus, and one time out of 1,000, that gene would make it across, and you'd get a plant that had some new feature to it. So it was laborious, it was expensive, it was unpredictable, but they did it. And today, we have what's called genetically modified organisms, and that's how that was done. So there's an upside to doing all of this stuff. That's a watermelon from the 17th century, and this is a watermelon from today. Now this was not created with natural selection, right? This is a genetically modified organism. It's a crossbred organism. It's a selectively bred organism that's taken us a long time, hundreds of years, to get from there to here. But you can feed a lot more people with this than you can with that. Look at that for a minute. We've been cultivating this for 8,000 to 10,000 years. And some say it's the result of intelligent design, and it is. We've intelligently designed the banana to be something a lot more desirable than it used to be. We did this. Can you tell me what that is? Anybody know?
Pumpkin?
It's an eggplant.
Oh.
Right? That's the way an eggplant used to look. And we've done all of this without CRISPR, Cas9. So we've been genetically engineering plants. Well, now we're doing it in a big way. This is a graphic from Anti-GMO Group. And what they're pointing out here is that half of the arable farmland in the United States is now planted with genetically modified organisms. This says 88% of your corn is genetically modified. And I don't know if you realize it or not, but corn is in everything. 93% of canola, 95% of sugar beets, genetically modified. And to add insult to injury, if you're an anti-GMO person, now DuPont has decided that they're going to use CRISPR-Cas9 to modify corn and wheat to make it increase its yield by 10 to 15%. And in the spring of this year, it's going to get planted out in the field for field trials, and in 5 or 10 years, it's going to end up on your plate. So if you're anti-GMO, this should really freak you out. But could the planet support the number of humans and animals that live on the planet if we weren't doing this? That's a rhetorical question, because the answer is no. Well, how about animals? If you're as old as I am, you watched the Little Rascals when you were a kid, and they had a dog named Petey. Anybody here remember Petey? Right? Had the circle around his eye. Petey was a pit bull. That's Petey. He was a pit bull. Pit bulls don't look like that anymore. Now they look like this. From the time I was a kid watching a black and white movie, they went from that to that. Isn't that amazing? That's a boxer, and this is a bulldog. So how did this happen? Well, the way this happened was the modern purebred dog, let's say, is only about 300 years old. Dogs were not bred for specific skills and specific looks until about 300 years ago. And most purebred dogs that we see today, like these, are only 150 years old. So in 150 years, we've taken random dogness and started to tune it into very specific looks and behavior sets. I have an English Golden, and I bought him as a purebred, and you wouldn't believe, out of the box, he was a retriever. He would bring me socks all day long. Now he brings me the paper. And you know, he just knew how to do it. Really amazing. So the way this was directed was because of dog shows, right? You go to a dog show, and the judge goes over to the pit bull, and he feels his head, and he says, okay, these bones are nice, but these bones on this pit bull are nicer. The shape of his head is more of the ideal pit bull shape. And then all the breeders who breed pit bulls say, wow, we got to breed for that shape if we're going to win the prize. And it doesn't take too many generations of doing that before you go from this to this. Now, there is an upside to that because you get things like a great retriever or a great bird dog, but there's also a downside. Because look what we did to the poor bulldog. They can't mate naturally. And they can only give birth through cesarean section because of what we did to the bulldog. We created a creature for which there is no natural environment, and if we let it out into the wild, natural selection would snuff it like that because it just can't continue. The 4 rules I gave you about natural selection, one of them was we had to be able to procreate. Okay, end of story. So there's a downside to doing this kind of thing, but there's also an upside. Look what it did for the chicken. Since 1950, we've produced chickens that produce 80% more meat with the same amount of feed. And this is through selective breeding primarily. Just, you know, breed chickens that grow faster and grow fatter. Last year, though, we started to use CRISPR-Cas9 to genetically tweak animals directly. And we patented the first Aqua Advantage salmon. This is a patented animal that's been genetically geared using CRISPR-Cas9. Now, if you're a salmon farmer, this thing is great because in, 18 months, where a normal salmon would grow this big, the Aqua Advantage trademark salmon would grow this big. So that's a good deal if you're a salmon farmer because for the same amount of time and the same amount of feed, you get a lot more salmon meat to sell, and that improves your profitability. Now, one of the things they can do, since you're genetically modifying this thing in the first place, is you can do things like induce sterility, which is probably pretty smart, because there's a huge downside to doing this. If that salmon were to get out, it could escape and reproduce with the wild salmon and modify what's going on in that environmental niche in ways that are not predictable. Because you don't know what would happen if it took over that environmental niche. You don't even know if it would take over that environmental niche. Maybe it'd be too slow to get away from the bears. Who knows? Maybe it wouldn't be able to swim far enough upstream to mate. Maybe it wouldn't know how to swim upstream. So, might be unintended consequences, might not. Another thing that can happen is it can reproduce with brown trout. Would it pass on that huge growth spurt to brown trout? And could the brown trout mate with something else, a third species, and they get real big? Would it be possible to walk across the river on the backs of big fish? You know? So you have to be considerate of all the things that might happen, but don't let that slow you down. This is what's going on right now in terms of modifying, using CRISPR-Cas9 technology, animals that we depend on. They're growing cattle without horns. Who cares? Well, if you were a farmer of beef cattle, you might care because cattle with horns can hurt each other. And there go your profits, right? If you're tending to wounded animals. And they can also hurt you, and there go your profits if you're tending to getting horned in the side. So what farmers used to do is they used to literally saw the horns off their calves and burn them at the root so they wouldn't grow back. And this is a pain in the neck for the cattle, and it's a pain in the neck for the farmer. So why not just modify them so they don't grow horns? Why not? Some other things that are going on. Pigs get a disease called swine flu. And remember, swine flu is a virus. And a virus, when it goes into the body of an animal, it disrupts the cellular structure and steals components out of the cellular structure to build copies of itself. That's how viruses propagate. So when they infect a pig, they do a lot of damage to the pig's chromosome. So one of the big experiments in 2015 was they went in one shot with CRISPR-Cas9, they repaired 62 pig genes, one shot, and it was 100% effective. They proved in doing that just how effective and efficient CRISPR-Cas9 technology is, and by doing this also, they can make the pig resistant in the future to ever getting swine flu again. Now this has all been kept in the laboratory so far. None of this has made it into the outside world, but plans are there. In China, they're building a factory where they're going to produce 1 million beef cattle a year. And they will be genetically modified and specifically bred to create more lean meats that taste better than a normal cow would produce, a normal bull. So everybody will be able to eat Kobe beef in the future, right? Without all of the expense of the Kobe beef. And you know, if you read this, you'll see lots of examples of things. Chickens that produce only females. One of the big tragedies of the world today is if you go to a chicken farm, what you'll see is they hatch lots and lots of chicklets and chicks, and there are people that do nothing but stand there and find the males and kill them, because males are useless to them. They don't lay eggs. So why don't we just produce chickens that only have females? Then we don't have to kill them off. It's a humanitarian thing, right? So moving on to humans. We have the technology now to implant new hands, to surgically attach new hands to somebody who has lost their hands for one reason or another, an accident, a birth defect, disease. This kid got 2 hands attached to him, and you know, they work. He's learning how to use them. He's got dexterity. It's a marvelous thing. Does anybody think this is a bad thing to do? Show of hands. It's a bad thing to do? It's a good thing to do, right? How could you say this is bad? Problem is, where do you get the hands? What if you needed an ear and you could grow it on the back of a mouse, and then you could cut it off the mouse, and you would have the cartilage now that was an exact copy of the cartilage in your ear, and then you could use some of these techniques we've talked about, genetic modification of stem cells to turn them into skin cells that were your skin cells, put them on this scaffold, and then surgically attach it to your head. And you've got a new ear, right? So this is a good thing, maybe. An animal that is part human and part animal is called a chimera, or if it's part one kind of animal and another kind of animal, it's a chimera. This is from an old Greek myth. But this kind of work has been going on for a long time, too. There's one I wanted to talk about. There's, oh, this one. In China, they did an experiment where they produced goats that didn't give goat milk, they gave human milk. You snicker. But consider this. You have a baby and you want that baby to have human milk, you want that baby to be breastfed, but you're the vice president of Monsanto and you just don't have time for this stuff. Today what people do is they give their kids formula, and clearly formula is not as good as human milk would be. So you could get one of these human milk producing goats, and you can milk that thing, and you can put it in bottles, and then when your baby needs milk, you've got human milk in a bottle that you bought at ShopRite. So again, potentially terrific. And there are a lot of other things that they've done. You know, they're trying to find cures for Alzheimer and Parkinson's, and there was another interesting up here. Oh yeah, pigs with human blood. That would be a cool thing. You need a transfusion, and you need some blood, and you want it to be the right RH factor, and you want to have an unlimited supply of it. Why not grow pigs with human blood of all the different types, and then when you need it, you've got it. Moving into the future, again, this is all laboratory right now, but in a couple of years, there's a very bad disease called limbal stem cell deficiency. It's an eye disease and it causes blindness. It's very painful. About 300,000 people get this a year. And it's curable using this technology. What they do is they take, it's a stem cell deficiency. So what they do is they take stem cells from your body, use CRISPR-Cas9 to modify them into the stem cells for your eye, and it's for the outer coating of the eye. And then inject those modified stem cells back into the eye. They find where they belong, and they replace the bad tissue with new good tissue, and the blindness goes away. Okay, this has been done in the laboratory, and it's something that they're hoping will be a viable cure in the near future. They can also find out you have some kind of cancer, some kind of tumor, and then they can modify immune cells to specifically target that specific disease and potentially cure it. That's another thing they're working on. There's a disease called Leber congenital amaurosis, and they're going to use that in the real world in 2017 to cure this disease. In August, last August, using these technologies, they produced islet cells from the cells of Wendy Peacock. They genetically modified them, and then they put them back into her pancreas. And cured her type 1 diabetes. She no longer has to take insulin. So here's a case where they've used this technology in a way where, you know, it can't escape into the wild, it can't be passed on to her children, but it can save Wendy Peacock from being a diabetic. Here she is 43 years old. All her life she's been taking insulin, and now she no longer has to. So huge potential on the upside for doing these kinds of things. Here's something we were talking about at dinner last night, right? Mitochondrial disease in Great Britain was seen to be quite a serious problem. Now, there's mitochondrial DNA that's passed on from the mother's side of the family, all right? But it's a genetic disorder. Mitochondrial disease is. It's always transmitted, passed on generation to generation. It's a genetic disorder, and what it does is it affects the mitochondria inside the cells, which is used to produce energy at the cellular level. So if you have mitochondrial disease, you got it from your mother, and it makes you very weak, it makes you suffer a painful disease, and it may even kill you. And if you have children, you're going to pass it on. So in Great Britain, they realized that they could actually cure this disease. And the way they do it is this. If you have mitochondrial disease, you can go to a doctor and you say, I want to have a child with in vitro fertilization. The doctor finds a woman who does not have mitochondrial disease. They get a sample of her mitochondrial DNA and replace yours with it. Now we've got an egg that's got no mitochondrial disease in it, but it's got somebody else's DNA in it. So then, you know, they do the IVF thing, they implant it, it grows into a child who does not have mitochondrial disease, but has 3 biological parents, which is new and unique. And they've destroyed the disease from being passed on. It's not going to be passed on anymore. That's the end of mitochondrial disease in Great Britain, as long as people take advantage of this capability. Now, you can imagine that when this was brought up, there was a lot of controversy about this. Oh, you know, we're really getting into the weeds now. So they took it to Parliament. There was a lot of discussion. There was a vote. And they came to the conclusion that the risks were small, and helping people carrying certain diseases to have healthy children mattered more than rather formless worries about playing God. So they went ahead and did it. So now it's legal in Great Britain. In 2016, the same idea was brought forward in the US because of the success of what happened in Britain. The National Academy of Medicine went to the FDA and said, can we replicate this procedure in the US? And they got a lot of pushback. So if you have this disease and you want it cured today, your insurance company will not pay for that cure, and you would have to fly to Great Britain and have it done, but you could have it done. Now, we recognize somewhere around 6,000 diseases that are caused by mutations in DNA or mistakes in the DNA. Most of them have no treatment. None of them are curable using traditional methods. And they're diseases like cystic fibrosis, muscular dystrophy, sickle cell anemia. These have all been cured in laboratory animals using CRISPR-Cas9 technology. So we never could fix these things before, but we can fix them now. We could eradicate hereditary diseases and vulnerabilities. We can also enhance abilities. And this is where we start getting into the real weeds, right? Do we want to start doing this? Other things we can do is we can extend normal lifespan. Right now we have the technology available, and it will all be in the market in 5 to 10 years, mind you, to extend your lifespan typically to 120, 150 years. And you would be healthy and relatively feeling good the whole time. That's pretty cool. I'm looking forward to that one. And we can even re-engineer humans. And you know, there are things about humans that are not exactly perfect, right? Everybody's got wisdom teeth problems. And the reason for that is because we domesticated ourselves. Our jaws used to be bigger. Well, now they're smaller because of our self-domestication, right? We got involved in modifying our own evolution, made our heads smaller, got more crowded teeth, and now when you get to be 18 years old, somebody's going to pull your wisdom teeth out. Well, why do we have to deal with that? If we can have cattle without horns, why can't we have faces without wisdom teeth in them? Right? So that's a possibility now. So who's thinking about all of this? Last December, the International Summit took place in New York. I think it was 3 days. It included the Academy of Sciences, the Chinese Academy of Sciences, the UK Royal Society, and they came to a conclusion. They talked about all of these issues. They're scientists, they're thoughtful people, and they understand the technology. And they said human genome editing is ethical and appropriate as long as it is not done for reproductive purposes. And what that meant was you can go into the lab and you can modify a human egg. You can let it turn into a blastocyst, but that's as far as it goes. After that, you've got to destroy it, because when it starts to turn into a human being, you're not allowed to do this. But that was just a suggestion. That's not the law. In many countries, though, including many in Europe, there are laws that forbid human germline modification. So even doing that business with mitochondrial DNA is going to be illegal in some places. The National Institute of Health won't pay for any kind of research that relates to this, but there's no law against using private funds. So guys like Craig Venter, they're still working on this. Monsanto is still working on this. DuPont is still working on this. Money is being spent. Lots of money is being spent. China doesn't prohibit any of it. They've actually modified the human genome and taken it a lot further than we have with allowing babies to be born in some of their testing work. Let's go on. Let's do some thought experiments now. This is where the fun starts. So you decide you want to have a baby, but you decide you want that baby to be free of any genetic defects. So you go to the doctor and you say, Doc, I want to have a baby, but we're going to do it because it's Valentine's Day using in vitro fertilization. And before we put those eggs and sperm together, we want you to test them and make sure that there are no diseases. And if you find any eggs or sperm with disease potential, and then we're going to reject those, only use the good stuff. Now, show of hands, is that okay? Is that okay? Wow, you guys are sticklers. How about this one? You go for the in vitro fertilization, and you say, well, we're not going to get a donor this time, we're going to use our own stuff, and we're just going to screen for diseases. Is that okay? Show of hands. Okay, you're a little better with this one. Good. How about this? You go to the doctor and you say, and while you're looking for diseases, also check to see, I don't want a kid that's too short, too tall, too smart, too stupid, too anything, you know? I don't want her hair to be too curly, Is it okay to do that? Does it start? Alex is ready. Take note. How about this? Doc, I want my kid to have blue eyes. Is that okay? Show of hands. I got one. Hey, surprisingly, 4 people. Okay, you go to the doctor and you say, Doc, I would love to have children, but I have the BRCA1 gene mutation that yields a very high probability of breast cancer. So I'm not going to have children. And he says, well, you know what? Modern science today, for about $1,000 or maybe $10,000, whatever the price is going to be, I can fix it so you can have babies who do not have the BRCA1 mutation. So you can have kids, and they won't get breast cancer. Is that okay? Show of hands. Okay, so eye color bad, no breast cancer good. That's good. How about this? You go to the doctor and you say, Doc, I want to have kids, but they've got to be athletes. They've got to be good at what they do. I don't want the other kids making fun of them. And he says, not a problem, $1,000. They're going to be pretty normal. You know, nothing great, but they'll be okay. Show of hands, it's okay? Not that many athlete fans in here. How about you go there and you say, Doc, I want my kid to run the 4-minute mile in a minute and a half. And he says, I can do that. Is that okay? Show of hands. Wow, 1. We got 1. 2. We got 2. Now we get serious. So remember, you can do something that affects only the individual, depending on when you do it and where you do it. You could make this kid an Olympic athlete, and it would only be him. But you could also modify his genetic line so that 50% of his kids could be real athletes, or you could do it with the gene drive, and every descendant that kid has is going to be an Olympic athlete, right? So that's a possibility. How many people think that's okay? Oh, we got 3 now, so you're starting to buy in. Okay, here's another one. Suppose you go to the doctor and you say, Doctor, I don't want any of my descendants to ever have Alzheimer's disease. Should you do that? Is that okay? So you guys don't like diseases, do you? Now suppose you go to Congress, you write your congressman and you say, you know, we can cure Alzheimer's disease for all Americans. All we have to do is guarantee that from now on, every child that's born in this country has an anti-Alzheimer modification, and it's attached to the gene drive. And all of their descendants will not have to worry about Alzheimer's. And after a few generations, there will not be an American left who has to worry about Alzheimer's. And Congress says, hey, good idea, let's pass a law and make that a law. Is that okay? Show of hands. Okay, that's something to think about. What if the same thing happened? You said, you know what? We Americans are exceptional people. And we deserve to be exceptional people. And we should all have IQs north of 160 and maybe up to 200. So everybody in America could be smarter than Einstein. And we can make it happen. You know, not today, but give us 5-10 years. This is something that we could possibly do. Should we pass a law to make every American have an IQ of 200? Show of hands. So you're anti-disease. Okay, we got two. Okay. Now you're Donald Trump. And you know that this is all going to be illegal in the U.S. And you say, well, I don't care, I'm Donald Trump. I'll go to China. I can afford it. I don't have to worry about insurance paying for this. I got the money. I've got, what has he got, $10 billion, he says? Then he gets on a plane. He goes to China, and he goes and he has a consultation with the doctor, and he says, Doc, I want you to modify some genes in my sperm and my wife's eggs, and I want you to attach them to the gene drive, these modifications. All my kids are, from now on, they're going to look like me, they're going to have hair like me, they're going to be smart like me, and all of our descendants from now until the sun hits the earth are going to be little Donald Trumps. That can happen, right? Because he's got the money. He could make it happen. And you, because you don't have the money and it's illegal in this country, couldn't. So, this is a technology that could become available only to the rich. So now we get to the end of my presentation, and we're back to the questions that we started with. So there's a lot of things to discuss, and I'll bet some of you guys have opinions. Who has an opinion or a question or a comment to make? We've been a lot stricter than Europe so far in some things. They're stricter than we are about GMOs, but we've been stricter than they are about genetic engineering. So, you know, it's a mix, toss-up.
Ron, thanks. That was a terrific presentation. Thank you. Two things came to mind when you were asking the questions. One was that I want another category, which would be, we don't know yet. Besides yes, yes, do it, no, don't do it. Or the third category, I don't know yet. Because we don't know what the risks are, really. And so part of it would involve evaluating the risks before I could answer the question. And the analogy that came to mind was, let's say that 100 years ago, when we first started using fossil fuels and building automobiles, not realizing what would happen to the climate. And let's just say bad things happen to the climate. We'll just make that assumption. Should we still start using fossil fuels? So it's I find that analogy to be a really interesting question, because we've gotten a lot of benefits to date from using fossil fuels, and they've allowed us to invent solar cells and fusion energy and so on. So, there's also the question of if you do something in the short term that might have a lot of risks with it, but it might bring great benefits in the long term. So I don't have any answers to those questions, but just a thought.
Yeah, neither do I. The reason I brought all that stuff up, I mean, these think experiments that we were doing, that's probably the first time you've ever been faced with that question at all, right? Because up until now, we didn't have the capabilities to do that, and maybe you would see something in a science fiction movie, but now we're talking about reality here. And we're getting to the point where we have to make decisions about this, and if we don't understand what we can do, we don't understand how it's done, if our congressmen and senators aren't educated, they're going to make bad decisions. Maybe they'll be driven by people with money. So we have to be very, very careful about how we think about these things. We've got to make sure we're educated.
So I'm going to ask a question myself. One of the things that when we talk about...
do that.
Yeah. We were talking about what the law should permit, but the law is not a very effective instrument at molding what's going to happen here. The law is good at tamping down at on things that we don't like. We don't like crime of various sorts. We don't like people behaving badly, but people still do. There are still murders. There are still robberies.
There may be a gene for that, you know.
Whereas, but with something like genetics, one person or can break the law, do something that will get them put in prison, and maybe we find out later and we put them in prison, but meanwhile, they've released a trait into a population that there's no turning back. So it's not like Congress is going to be able to fully control or shape how this technology is going to
Well, it
develop.
depends on whether Obama proposes it or not.
And the other thing is that I should point out is when there's a law against something, you tend to get only the bad consequences of whatever that thing is, and you don't get many of the benefits.
Right. Well, like I said, this is an easy thing to do. All the scientists agree. It's an easy thing to do. I heard an interview. I was in the car on Friday, and the guy who did the work on the Anopheles mosquito that I was talking about was being interviewed. And they asked him, you know, how long did it take you to genetically modify the Anopheles mosquito to not be able to carry malaria? It took him 2 weeks. It took him 2 weeks. So we make it against the law here, say, you know, no genetic modifications on anything ever again. So, you know, you go to China and you get done what you want to get done in China. Or maybe suddenly all the South Koreans start winning the Olympics, you know. What's going on here?
Is there a worry here? I mean, you kind of touched upon it, that certain countries are going to allow this to happen and others aren't because of their morals.
That's the way it is right now, yes.
Yeah, so does this mean that 100 years from now there's going to be one country that, oh, surprise, we've got super troopers, we're going to invade?
Well, it would take 100
whatever.
years. It
Well,
might be,
whatever,
Yeah.
like, you know, the hypothetical.
well, and there's another consideration, right? Well, what does the military think about all this stuff? Are they going to start thinking about super soldiers?
Right, so.
You know, so maybe you can't do it, but maybe if you join the Marines, it could be done to you.
Yeah, so that question about, you know, oh, hey, go to the government and say you can wipe out Alzheimer's, right?
Unless we grow our own food, there's no way we can control when we go to the store because if there's no labeling on GMO produce at this point.
That's right.
And I don't, so we're all guinea pigs or at the mercy of the people that produce these items
Yeah, and
for
they
our
will-
consumption.
They have promised me that there are no side effects to GMOs. But yeah, you're right. There's no law in this country that GMOs have to be labeled, and they're not. And you know, Monsanto and DuPont do not want them labeled because they're afraid that people will stop buying their products. You know? So yeah, I understand their point of view.
Thank you for your excellent presentation, Ron. Are brains continuing to get smaller?
Well, are we fully domesticated yet? What do you think? I don't know. We'll know in 100 years. We'll look back and say, hey, look, our brains are smaller now than they were 100 years ago. They haven't shrunk since that. Since about 20,000 years ago, they started to get smaller in size, and then it stopped. But the last mutation to the brain was only about 10,000 years ago. And you know, in geological terms, that's bang, zoom, 10,000 years? We mutated just 10,000 years ago? We could mutate tomorrow. But, like I said, would it be selected for if it happened now? You know, is there any reason why there would be a population sweep if your kids suddenly had IQs of 300, you know, yeah, they benefit from it, but it's not something that would overtake the population because there's no pressure anymore to have to be smart, right? Like they used to.
Plus the population is so large.
Yeah, yeah. How do you penetrate a population of 7 billion people?
Thank you for your great presentation. I'm over here.
Yeah.
I think I understood most of it. Well, no, maybe 20% of it.
Well, you can have the slides and read yourself to sleep tonight.
But the thing is, I am a sociologist, and you know, whenever I think about the progress made by science and technology, I always have to put it in the context of society, of social, economic, political power, forces. And I think how the consequences of these would depend so much on those factors. Who has the power? Who has the money? Which country, can set the standard and all of that. So I think that is something which needs to be taken into consideration. And in a situation where everybody really can come from the same, have the same chances of taking benefit from all this research, I think it would be something more acceptable than the way it is today, where some can benefit more and can enhance their power, and by the same token, have quite negative implications for the rest of the world. I'm thinking of Donald Trump.
Well, it hasn't happened yet, right? I mean, we're there, but we still have maybe 3 or 4 years before we have to answer the question. Not a lot of time, but we still have 3 or 4 years before we're really faced with it. Yeah.
2 things. I'm pessimistic. I think that there is no stopping this. No law is going to stop these changes that we are now being able to do. And you did speak about one way this can be used is to help with diseases, to make people live longer and also be healthier. But we have a problem. We have too many people in this world. And the big change in increase in people in the world population happened just about the time that medical science was able to impact health in the 1800s, early 1900s.
Yep.
So 2 things. One, if this technology allows more people to be born and allows them to last longer, how many people can this earth handle before we start going into real negative territory?
Yeah, good question.
Yeah, I have a shorter question. What do the creationists think about all this?
You'd have to ask a creationist. I don't know. Do we have any creationists? Are you
They left early. And I have another question. What's a futurist?
A futurist? Well, there are a bunch of futurists around. The most famous futurist today is probably Ray Kurzweil. And Ray Kurzweil discovered about 40 years ago that there are some things that can be predicted with a great deal of accuracy. And he took up a study of what those things are and identified one specifically, which was artificial intelligence. And he started tracking it, and all of his predictions so far have proved right on the nose. And so he's predicting the future very accurately, and it's coming true just as he said it would. He's a futurist.
What did you predict?
What did you predict?
That's my next talk.
Ron, thank you for a great presentation. I'm here. Thinking about this ethical side, just something which is coming to my mind, I'd like to ask you about your comment about this. Once there are so many opportunities, okay, what do you think if at this stage such legal principle could be accepted as avoiding unnecessary risk, okay? Which means once all those things may carry risk, fighting disease is something which has priority, but producing athletes not necessarily according to this principle. What do you think about it?
Well, that's what happened with the mitochondrial DNA in the UK. They said there may be risks associated with this, but the disease is so bad that we can't even worry about that stuff. It's much more important to get rid of mitochondrial disease, period. And so that was the decision they made and the direction they took. And that's a decision that's going to impact future generations, right? So that was a big one. And we, on the other hand, were presented with the same one, and we said, no way, we're not doing it. There are going to be meetings, they are going to keep discussing it, but right now the answer is no. So yeah, you know, when I heard that scientist interviewed on Friday, one of the things I kept thinking was, wow, this guy is really smart and he's right on. When he decided to test that mosquito, he took it to an island. So there were no risks that he couldn't overcome. He was really good about it. What I worry about is congressmen and senators, because these are guys who are faced with decisions like this, and you know, they carry snowballs into Congress and say there's no global warming, because I have a snowball. So there, you know. Do I want that guy making this decision? He's the head of the science committee. Oh my God. You know? But we got what we got. So I think mistakes will be made. I'm a little pessimistic too, but I also think we're in a foot race, because there are forces at work in the military, in the government, among the rich. You know, don't forget, there are people in this world who own half of the planet. There are, what is it, 85 people? There are 85 people in the world who own half of the planet. Those guys can do anything they want.
And do.
And you can't stop them. I don't care what laws you make, they own the government, right? So if they decide this is going to happen, or it's not going to happen, or it's only going to happen for them and their friends, that's what's going to happen. So, you know, it's good to be educated, though. I mean, we try.
Something just reminds me of Arthur C. Clarke wrote Childhood's End.
Yeah.
And sometimes I look at the kids now, and they're different. Their brains are very technological. And when did that happen? I'm stupid. I don't know when. You know, I know they work the computer, but not that iPad stuff.
Yeah.
So
That's a different thing, though. That's called plasticity, right? The brain is shaped, and the brain is unique in this. The brain is shaped by what it does. The brain reorganizes itself based on what it's presented with. So as a very young child, if you had been presented with an iPad, your brain would have shaped itself around that paradigm, and you would be able to operate an iPad no problem. It would be second nature to you because your brain is designed to do it. Your brain, unfortunately, was presented with Life magazine, so you've got to turn pages, and I understand that. It's a handicap, but you'll and you know, in 20 years, kids will be doing stuff that we'll look at and we won't even know what they're doing. I mean, there's stuff going on right now, I don't know what it is. I don't know what Pinterest is. It's happening, but I don't know anything about it.
My question is on the current U.S. law. You said that people are allowed to experiment on human cells, and
Yeah.
as long as they don't allow them to grow past plasticist stage.
Right.
What's the penalty if you do allow them to grow further, and what are the legal consequences for the organism that grows to become, you know, an independent living creature?
Gee, I don't know. We haven't been faced with anything like that yet. It'll be interesting to see which way the Supreme Court goes, because they're going to be faced with some really tough questions in the very near future. Because, you know, when you get to questions at this level, people tend to run toward documents that have been around for a long time and say, well, what does the Constitution say about doing this? You know, is this life, liberty, and the pursuit of happiness? Is this the right to bear arms? Is this freedom from, you know, where in all the documentation does this live? And then the Supreme Court says, well, we'll interpret that for you. And, you know, that hasn't happened yet. It's going to happen in the very near future. Get ready.
I'm a little confused about this the bias against the genome and the genome of the alteration of whatever party that people seem to have a bias against it. I mean, We didn't notice that bias when they invented fossil fuels. We didn't know then, like we know now, that it would destroy the ozone layer or something, and perhaps we would have not used it to propel automobiles at the time.
That's not true. There was a lot of bias against fossil fuels when they first came up. There were people that said you can't survive in a train that goes faster than 30 miles an hour because you won't be able to breathe.
So then it's my question. Is that because people are just generally reactionary? They just resist progress and change? That's my question.
I think this is a case of fear more than anything else. People are afraid of what they don't understand, and not a lot of people understand this. Most people have never been presented or would even come to a presentation like this. I had to do months of research to gather all this material together, and it was changing every day. I added to a slide this morning because something occurred to me that I hadn't researched, and I had to go look it up and add it to a slide. It's a lot of work. And if you're not willing to do that work, then you chose to be ignorant. And when you choose to be ignorant, then you have to lean on where are my comfort levels and where are my fears. And everybody's afraid of what they don't understand, so they say, let's not go there. Like that's going to work. It ain't going to work.
So I'm wondering, I'm sure you're familiar with GMO, Roundup Ready corn and stuff like that. And I think recently California came out declaring that Roundup was carcinogenic or something like that. So this is kind of like we developed the technology. It's now 90% of all corn or whatever. And now we're finding out some of the side effects of it, like the cancer-causing Roundup Ready that's all over the soil and everything. So, just wondering what your thoughts are, on that whole process.
Yeah, what he's talking about is Monsanto came out with this weed killer, Roundup, which is very powerful. And the downside of it was it also killed the corn. So then they genetically modified the corn to be resistant to the Roundup. That means they can spray Roundup and kill everything that's there except the corn. Which, you say, hey, terrific. The bad news is, though, that now you've got corn that's covered in Roundup, and it's absorbed it, and it's in the soil, and it's going to be there for a long, long time. And you're getting exposed to it. So you're eating a GMO plant, and you've been assured that the GMO plant, the modifications to the plant, will not harm you. What they don't tell you is it's saturated in Roundup. And that's carcinogenic, So yeah, I've got a problem with that. I don't know if they can clean that stuff up or get it out of the corn or whatever, but I am in favor of labeling GMOs for that reason. I don't care that they modified the corn. I'm okay with that. What I don't like is they're saturating it in a poison that now I'm forced to eat, and it's in everything. Oh my God.
I would slightly disagree that it's not about lack of understanding that we have these fears, but for a lot of people in this room, we remember thalidomide babies.
Oh yeah.
And what happens, when you do these, I don't know what's going to happen experiments.
Well, and that's why we have the FDA, and that's, and when Congress says we don't want to fund the FDA, we want to get rid of them, that's a mistake because they're protecting us from that ever happening again. And yeah, the FDA should be involved in this, and they should be educated, and they should be talking to the right people, and they should be talking to us. Because when decisions are made behind locked doors or in rooms where we're not allowed to go, I think that's a bad thing. Because it comes back to bite us in the end. And who got paid off over that whole thalidomide thing? And who went to jail?
Okay, well, thank you very much. That brings the presentation.